Poisonous snakes from all over the United States and 11 foreign countries are kept at the Ross Allen Institute in Florida. Curator Andy Koukoulis is an expert at handling them. But before going to work, he puts on his snake boots. These are made from extra heavy leather, which the rattler's fangs cannot penetrate. Grabbing his snake hook, Andy is ready to milk some of the most poisonous snakes in the world, including the largest of them all, a huge king cobra. This lab is the country's top producer of dried snake venom for medical purposes, and rattlers are one of their main sources. There is no safe, easy way to obtain their venom. Each snake must be milked by hand. Someone must take the chance of being struck by the rattler's 3/4-inch fangs. Someone like Andy. Wild snakes never become friendly, never cease trying to strike their captors. Handlers such as Andy can never let their guard down. Expert fingers press the venom glands as the deadly substance oozes out through the sharp fangs. Even after milking, there's enough venom in the glands to cause serious poisoning. When a rattler actually strikes, the lower jaw drops and the head tilts backwards. The fangs swing forward and the mouth opens at an angle of almost 180 degrees. As the fangs dig into the victim, the muscles surrounding the large glands in the snake's head contract and force the venom into the wound. The trick here in the laboratory is to get the venom into a beaker and not into a wound. This procedure requires scientific knowledge and nerves of steel. Baby eastern diamondbacks proportionately have larger amounts of venom than the adults. Pound for pound, it is among the deadliest snakes on Earth. The venom is ejected from a hole in the fang, much like a hypodermic needle. Expertly, Andy returns the snake to its box. Step 1, the extraction is completed. One of the main uses for venom is the manufacture of anti-venom or anti-snake bite serum for treatment of bites by pit vipers. Many processes are involved in the development of this serum. Bill Gleason transfers the raw venom into the jars, which are capped and placed in special freezers. These are the initial steps in a process which, when completed, will produce an anti-venom that remains potent for at least 5 years and can be carried in the field. It is available only by prescription and should be administered only by a doctor. Next, Bill prepares the frozen venom to be dried by vacuum dehydration. This is the same basic process as that used in making freeze-dried coffee. With the addition of water, prompt injection of the antivenin prevents death, relieves pain and other symptoms, and shortens convalescence from snake bite. It is never too late to administer anti-snake bite serum, and many lives have been saved by the injection of large doses, even when the victims were apparently dying. Prompt first aid with a snake bite kit will usually save the victim's life, but only antivenin can ensure saving both life and the injured limb. So each step is absolutely vital. What about research? This is what poisonous snake venom looks like under a microscope. Structurally complex, it has never been completely analyzed. Dr. Joseph F. Gennaro, a New York University professor and a consultant at the Institute, is an expert on venoms and probably knows as much about their effects on tissue as anyone alive. A simple experiment demonstrates the effects of venom on any living cell. Whole blood contains healthy red cells with round protecting membranes. When the blood is poisoned, the cell membranes are broken down and the hemoglobin released. Once the membranes are dissolved, the contents of the cells are digested and the blood turns white. This is basically the destructive process which Dr. Gennaro is demonstrating. For the victim, the results are more noticeable and dramatic than in a test tube. Hemorrhaging occurs, muscles deteriorate, the skin tissues blister, and vital organs are impaired. So tests like these will eventually save lives and relieve much human misery. His work, like hundreds of others in the study of poisonous snakes, goes unheralded but not unappreciated. Under a microscope, we are watching a simulation of what happens in the victim's body as the venom starts to break down human blood cells. In real life, it's a slow, painful process, which these men of science are attempting to understand and conquer. Dr. Gennaro, in fact, was instrumental in developing an anti-venom serum from the North American coral snake. The research and products which these men develop will ultimately cause the rattle of death to lose its sting. coral snake, despite its size, has the deadliest venom in North America. And the rhinoceros viper is but one of 40 species of poisonous snakes kept at the Institute for venom extraction and study. Experiments in reptile medicine have succeeded in keeping these surprisingly fragile animals healthy and keeping their venom-producing capacity at a maximum. The Malayan pit viper, for instance, contributes a venom component used in the prevention and treatment of abnormal blood clotting. In terms of human lives, its venom is priceless. Various venoms are used to relieve pain, coagulate blood, and treat bleeders. Many venoms are utilized as basic raw material in medical and biochemical research. There's no way of knowing what miraculous use for venom may be discovered next. What child's life may be saved by it, or who may be spared unnecessary suffering. But new breakthroughs in medical science using this substance will surely be made, providing man with a gift from an unlikely member of the animal world.